Yes. Boa tarde, gente. Bem-vindos. Muito obrigada. Hoje a gente tem o professor Roberto Salguero-Gómez. Ele se formou em Ciências Ambientais pela Universidade de Cádiz, na Espanha. Ele fez mestrado em Gestão de Recursos Naturais na Kingston University de Londres e doutorado em Ecologia, Evolução e Biodiversidade na Universidade de Pensilvânia. Lá ele examinou por que e como as plantas diminuem de tamanho, um fenômeno ecológico que era pouquíssimo estudado, e as implicações ecológicas e evolutivas desse encolhimento das plantas nos níveis modular, individual e populacional. Desde 2017, Roberto atua como professor associado em Ecologia no Departamento de Zoologia na Universidade de Oxford e como Tutorial Fellow em Ecologia no Pembroke College em Oxford também. Sua pesquisa atualmente abrange questões relacionadas à senescência em plantas e animais e relações entre características funcionais e estratégias demográficas. O objetivo final do Roberto é reunir várias abordagens biológicas para examinar os aspectos demográficos do ciclo de vida de um organismo. E para isso ele usa tanto campo como laboratório e também modelagem ecológica e biologia comparativa. Ele é fundador e coordenador da base de dados COMADRE, que é uma base de dados de matrizes de animais, e coordenador também do COMPADRE, que é a base de dados de plantas. Então, muito obrigada, Roberto. Desculpa. So, the talk will be in English, gente. Então, gente, a palestra vai ser em inglês, mas vocês podem enviar perguntas tanto em inglês como em português. Eu posso passar para ele em inglês ou em espanhol. Ele é espanhol também, então a gente se entende. So, Roberto, when you are ready, you can start. We are very glad to have you here today.
Thank you so much, Nuria. It's a pleasure to have been able to chat with you. Also, Nuria has been a really close and very nice collaborator of mine during the last years, and I'm very happy to be able to use her as if it were as a nexus for conversations on ecological and evolutionary topics with the ecological Brazilian community, which is an area that I hope to strengthen further into the coming years as well. So now, as per the science, understanding where species are, and perhaps more importantly, where species will be into the future, is arguably one of the most important questions that we ecologists and land practitioners have to face. This is ever more important in the face of climate change and direct human anthropic interactions with nature. Obviously, pollution, habitat fragmentation, land degradation, we are culprit for, and we need to understand how all of those actions that we inflict upon biodiversity, from which we depend, is going to drastically share and shape the ecosystem resources that nature has to provide to us humans. And before I put you all to sleep, I suppose that the main take-home message that I would like you all to walk away with is the fact that if you put together theory and data, in this case, life history theory, and large access, open access volumes of demographic data, one really is very well equipped to address really important questions, which have to do with what species are going to be the winners and the losers to climate change, and therefore how climate change might shape biodiversity. This is the core of one of the 3 research teams that I lead in my research group at the University of Oxford. And a lot of the inspiration that you will be seeing from what I'm presenting today is actually not coming from ecology. It's coming from a relatively older, perhaps more established, and definitely more predictive science, that of chemistry. Let me take you back to high school or even primary school. The first thing that you probably learn about the elements, the atomic elements, is that they are organized. That they're organized in a very specific manner, that of a 2-dimensional table. And that if you know for any one element how many protons, how many neutrons, how many electrons, and how many levels of electrons there are in a given atom, you can place the different species, quote unquote, on that table. You can place them, you can organize them, you can give them names. You can also predict the existence of some species between molybdenum and ruthenium. We can predict that there should be another species that has not been up until 1876 discovered yet, but soon it will appear. It's technetium. And we can also understand, based on the location of different species, whether helium combined with lithium, whether radon is a heavy metal or not. It's really important for us humans to be able to categorize before we aim to understand, and most importantly, before we predict. And that's what I would argue is one of the main priorities in ecological and evolutionary research, to predict. You cannot predict, however, what you cannot understand. And you cannot understand what you cannot put in boxes. So my way to understand, my way to box, and my way to predict is using life history traits and life history theory. Let me define very briefly what I mean by a life history trait. So a life history trait is any of the key events along the life of an organism that back in the day, before the advent of smartphone technology, your grandparents probably would have deemed important enough to take a photograph with an old camera and to go to the camera shop to deliver the case and to have that photo printed. Now, obviously what happens nowadays is that you can, with the help of one of these, you can take photos of anything, from food to your pets, whatever have you. But back in the day, before you had to spend the money on taking a photograph, you would think very carefully, is this worth me paying for that money? And the ways in which those photos will make it to the album of your grandparents is if they were deemed important enough. That is how I would try to explain to you what a life history trait is. When you're born, when you first go to school, when you're introduced to society formally as an adult in certain societies, such as in the Fiesta de Quinceañera, when you graduate to go on and get paid in your first highly paid job, if you decide to meet up, couple up, partner, call it whatever you call, marry, when that happens, when you first reproduce, if you choose to, how many offspring you choose to have, how much time elapses between reproductive events, in the case of humans, between baby and baby, if you choose to have more than one. Perhaps one of the key life history traits that we look forward to the most is when we retire. We humans have got a post-reproductive lifespan, and we also have got a post-working lifespan. And apparently back in the 1700s, it was also in vogue to take photos of the ultimate life history trait, which from very short-lived species, like C. elegans, to very long-lived species, like bristlecone pine, is inescapable, and that is death. So life history traits are any key event that shape and qualify your life cycle. What's important to keep in mind about these life history traits is that they're universal. We have them, we humans have them. Succulent plants, as the ones that I have in my office, also have them. The bacteria on your skin also have them. Lions and dandelions, anything across the tree of life, rely on key demographic processes that shape life history traits. And understanding the extremes of these life history traits really help us first define that two-dimensional space that we're taking inspiration from chemistry. You've got examples of extreme dormancy. Species pause their activities and they go into dormancy or torpor, etc., or hibernation. Got some examples here, such as some mice, and also the case of orchids. There are other species that circumpopulate and circumvents the globe twice every year, such as the Arctic species, bird species that you're seeing here, and also some other plant species that have got very long distance dispersal events, such as coconuts. When it comes to reproduction, some of the Guinness World Record holders of reproduction belong to corals and also to orchids. Orchid reproductive events can release 1 trillion seeds per reproductive events. And that number is actually quite equivalent to what a single colonial organism of coral can release overnight. We also need to think about biomass, how fast you grow and how quickly you develop. You've got two examples here of the current record Guinness holders of this category. The blue whale, which puts on, when it's an adult, 80 kilos. That's about the average weight of an average European human. 80 kilos per day. Let that sink in for a second, 80 kilos per day. And then, of course, if you've got some plant fanatics in the audience, you will be able to identify the species at the bottom. That's the bamboo. The records of growth in the plant kingdom at 0.9 meters per day. So that's almost 1 meter every day. And an equally important life history trait defined by vital rates is that of longevity. A couple of years ago, there was a new record in the animal kingdom, that of the Greenland shark, which in a conservative estimate ages up to 450 years before it dies. These individuals in this species gain adulthood at around 250 years. So put that in perspective and compare that to when you first become an adult around ages 16 to 18. And then in the plant kingdom, we've got some really long-lived species as well. The species there is the Bristle pine, which is the current record World Guinness holder of non-clonal organismal longevity at over 5,000 years. So as I usually tell my students when I explain just about how long-lived this creature can be, species of these individuals of this species were around before the Egyptian empire became an empire. Now, all of these extremes in life history traits are ultimately shaped and constrained by investments, energetic investments and physiological constraints onto development, reproduction, and survival. And we should be happy that there are such constraints, because if we didn't have trade-offs between these vital rate functions, then the Darwinian demon would have evolved and we would be taken over by a single species. We wouldn't be able to look outside of the window and see the vast range of species that we find across the tree of life. There'll be just one species. It will live forever or very long. It will grow very fast. It would achieve adulthood very early, and it would reproduce frequently and very intensively. Now, Darwinian demons don't exist. Perhaps you could argue the closest thing would be us humans, some species in the insect class, and also bacteria. But the fact remains that all vital rates are constrained through trade-offs. What's important to realize about these vital rates, survival, development, and reproduction, is that they really are the universal currency in addition to DNA and RNA at a genetic level. At the demographic level, they serve as an ideal means to compare and predict. They are the universal currency of demography. Any species, as I said before, from lions to dandelions, from humans to bacteria, have to invest to a certain extent in reproduction, in development, and in survival. That's a little bit about the theory, but really I couldn't just do justice enough to this. There's been a vast amount of contributions to the field of life history theory, going back to Cole, for instance, who asked the question, when is it selected for to reproduce more than once? That is, to be iteroperous. And when is it adaptive to just be semelparous? That means to reproduce once and then die shortly after or a bit longer after, like we humans can get to do sometimes. Walking on the shoulders of giants, standing on the shoulders of giants, as I mentioned before, Cole, MacArthur, Graham, Charnoff, who's actually my academic grandfather, and many other contemporary researchers who are very much still alive and kicking these days. From this collection of papers that I just mentioned before, and very much instigated by Steve Stearns, comes the first way to classify life history strategies. I need to take a second to define what a strategy is. A life history strategy is the combination of life history traits. So if a life history trait is being long-lived, and another one is being semelparous, a species that is both semelparous but lives long, like agaves, for instance, have a very characteristic and unique life history strategies, the unification, the convergence of different traits. Now, this main tenet put forward by Stearns as a hypothesis states that organisms across the tree of life are self-organized according to a continuum. This continuum is called the fast-slow continuum, and it categorizes life history strategies from species that live short grow fast and reproduce profusively, like copepods, to species that live really long, they grow really slow, and they reproduce rarely, like bristlecone pines, as you have in that example. Now, obviously, these theories that I mentioned before, going back to the 60s and 70s, were tested when we were not really data sufficient. So when these theories were put forward, we only had decent demographic data for mammals, and not even, and for some birds. So one of the big questions that I've been trying to pursue in the last decades is going back to your Ecology and Evolution 101 textbooks and ask the questions, now that we're a bit more data sufficient, do those theories still remain valid, or do we need to revisit them? One of the things that you realize here is that we started working away in this phylogeny from lions, just chosen species for which we happen to have a lot of demographic data. So again, data on survival, development, and reproduction. And we've ended up across the full tree of life, realizing that in fact, where lions sit in that phylogeny is a speck, is just a small region over here for which we know a decent amount, but we don't really, up until recently, know a lot for the demography of other species. And this is something that's an effort that I have led called COMPADRE and COMADRE are trying to fix or trying to improve. So COMPADRE and COMADRE are open access databases. You have the URL there and you're welcome to go there and explore. Below you've got some citations to the papers that introduce those two open access resources. What they have is demographic information. Demographic information in a stage structured manner. So demographers go to the field, they categorize individuals in a population, and they follow them through time. Or they make inferences that allow them to retroactively evaluate survival, growth, and reproduction. These two databases are now open access, and we're currently working on with the leadership from one of my PhD students, Sam Levin, that I co-advise, on two other databases that incorporate demographic data encapsulated in another format called IPMs, integrated projection models. And these should be going out live in the coming year or so. What's interesting about COMPADRE and COMADRE, as you can see here, is that we have got heaps of species, we've got heaps of studies, and we have got a decent taxonomic representation. This is a screenshot of the Google Earth populated GPS coordinates in those two databases. I invite you really to go into that website and scroll around and just out of curiosity, see how much demography is coming from Brazil, how much demography is coming from Latin America. What you will see is that we've really extended the offer and extended the repertoire of open access data in Latin America in general, but there's still a long ways to go. And that's a really important message because as a recent paper in Nature, Ecology, and Evolution showed just last year, where most extinctions are happening is in tropical ecosystems, of which I believe Brazil has a lot. So we need to understand more about the species that populate your country and other tropical countries before it's too late. This is just a zoom into the taxonomic and geographic repertoire that COMPADRE and COMADRE offer together. And it highlights again the fact that, yeah, we've got a half full glass of water or a half empty. There's still a lot of efforts that need to occur in order to populate Latin America and other regions like Asia and Africa a bit better. So if you're about to start your research, I highly encourage you to look into what's already been done demography-wise and maybe make a meaningful contribution from a complementary angle. The ways in which the demographic data are collected, as you can see here, that's actually me during my PhD back in the lower Jurassic. People go to the field. If you're dealing with a sessile species like plants, you put down your permanent plots and you follow them through time. And from that, you can infer survival, development, and reproduction. If you're dealing with creatures that move, like some animals, then you can do marine recapture experiments or other approaches that still allow you to come back to the same output. And that is that of a stage or an age discrete model from which you can retain the vital rate information that I described before. Now, these matrix population models have become really popular. They become really popular because the theory behind them are really robust, and because the utility that has been developed through our packages and courses is quite extensive, really. From both the theory and our packages, you can derive a lot of really meaningful biological concepts and entities, such as mean life expectancy of the population, rates of senescence, age and maturity, and other important quantities such as population growth rates, proxies to selection gradients, etc., etc. So back to the actual research, using this RH software, which is also open access and co-developed with other colleagues from Denmark, what I wanted to know is if, in a first instance, when you look at the plant kingdom, do they behave according to the predictions by Steve Stern? That is, single axis, where you have got short-lived species that grow really quickly and reproduce a lot on one end. The other extreme would be defined by species that grow really slowly, reproduce rarely, but live long. Or do we need to understand other axes of variation to really describe the full envelope of demographic possibilities in the plant kingdom? So what we did is for 416 species from COMPADRE, we derive quite a few life history traits. Generation time, we derive other proxies to investments on longevity, such as rate of senescence and age and maturity. By the way, this rate of senescence is the rate of senescence for survival, not for reproduction. I'll tell you more about that later. We also define 4 different moments for reproduction. How much, how often, how successful, and during how long. Those are the 4 different moments that you have there. And last but not least, we also evaluated how quickly individuals in a population can grow. And also, because these are plants, if they decrease in size, how quickly do they do so? Now, if you're used to thinking about mammals, for instance, or some birds, the term shrinkage might be a new one to you. Plants as heavily modular individuals can decrease in size, and they can do that through many hormonally regulated patterns, but one of them results in the shedding of modules, and if you shed a module, you become smaller. So it was a really important life history trait to be able to encapsulate in this analysis. And then the hope, as I said before, is to see if you put that in a reduction dimension analysis, do you just come out with a single axis of variation that explains the fast law continuum, or do you need more? What this approach allows us to do is to also get quite evolutionary. So at the backbone of this principal component analysis that I'm going to show you in a second, there's a phylogeny. And in this analysis, what we can do in one go is to evaluate the relative importance of phylogenetic ancestry versus the relative importance of environmental filtering in shaping the demographic trajectories of any one species. What you have there is bristlecone pine, Pinus longeva, and black pine, which is an invasive species in New Zealand, but it's actually original from Europe, Pinus nigra. So they're both pinuses, right? They both belong to the same genus. So you would expect them from a phylogenetic perspective, you would expect them to behave the same. But they live in different regions, and the one lives in a desert, the other one lives in a Mediterranean ecosystem. So if environmental filtering, hypothesis 2, is more important than phylogenetic ancestry, hypothesis 1, you'd expect the environment to matter more in terms of what a species can and cannot do. So this is the first result that I'm going to show you. I'm going to walk you through it, and you're going to be seeing quite a few PCAs today. So let me explain for a second what a PCA does, in case you're not familiar with it. A PCA, or a principal component analysis, is a dimension reduction statistical multivariate tool. And what it does is, if you have got a really rich data set with a lot of points, a lot of rows, maybe the species, and a lot of columns, a lot of variables that you're interested in, it evaluates the orientation in an n-dimensional space, n being as many variables as you have, to maximize the variance. It then evaluates how those variables load onto that axis or those axes of variation to come up with a biological interpretation in our case. And here what I'm showing you is the first 2 PCAs of a PCA that in fact had 1, 2, 3, 4, 5, 6, 7, 8, 9 life history traits. Just to remind you, gray means investment on longevity. Red is the different moments of reproduction. For instance, when do you start to reproduce, how intensively, for how long. This gamma here is growth, and this raw letter here is shrinkage, and then in black you've got generation time. So let me walk you through the main results. Just with labels on them, you've got longevity and growth. That's what would equivalent to the fast law continuum. This first axis of variation explains in 416 plant species, 34% of the variance. Now, going back to the prediction from classical work by Stern, 80% of variation was explained in the first analysis that was done only on mammals and birds. So obviously there's something ongoing with the plant kingdom, undeniably. And that has to do with the second axis that I'll explain in a second. But before we move on to that, let me explain the first axis. So each dot is one species, okay? You've got a species to the left that grow fast, and species to the right that don't grow fast, but have got high longevity and a low rate of senescence, as well as a high generation time. That is, it takes a long time for the individuals in the population to replace themselves, as opposed to species on the left that replace very quickly. So they are fast livers, and the other ones are slow livers, therefore the fast, slow continuum. Now, what is the second axis? Well, if we look at the top, all 4 moments of reproduction in red are aligned to the second axis, right? With the exception of this one, the intensity of reproduction, which is pretty much shared between PC1 and PC2. And then what you have here is the ability to decrease in size. So you have got species that reproduce intensively. They're highly iodoropterous. That's what that S means. They have a high net reproductive output, and they have long windows of reproduction, contrasted with species that have the propensity to decrease in size at the individual level quite frequently, and then they don't have the opposite, so they trade off, right? The way to explain this is that in plants, reproduction usually, but not always, scales with size. And so if you decrease in size, you're not as reproductive. But another way to think about this axis is you have got species that reproduce multiple times during their lifetimes at the top. So iodoropterous species, and species that don't reproduce much during their lifetime, so close to semelparous or strictly semelparous, and they can shrink at the bottom. Together, these two axes explain about 55% of the variation. So it's not even with two axes coming together to the 80% in mammals and birds. And so the main message that emerges from this analysis is that plants are way more complicated demographically than mammals and birds as evaluated in the past. Okay, another thing that we know from this analysis is that the signal for phylogeny was very weak. So the Pagel's lambda parameter, which ranges between 0 if phylogeny is not important and 1 if it is very important, was actually 0.25. So that implies that the phylogenetic relationships between each data points, each point in that graph, do not constrain what species can do. Instead, what we found is that the habitat where the species are based has a relatively greater effect in explaining this demographic variation. So we now have a way to classify plant life. Yay. We can, just like in chemistry, think about how many atoms, how many electrons and how many levels of electrons, right? So if you look from left to right, that would be how many electrons in the outermost layer. And if you think about the vertical axis, that would be how many layers of electrons, right? And here, all you're thinking about, though, is how fast or slow living you are and how much do you reproduce during your lifetime. Great way to classify life. Innovative in that it hadn't been considered before. Can I predict, though? And that became the big question in the research agenda of my group during the last years. And the answer is yes. What you can see here is each point, again, color-coded, corresponding to a species population, according to the growth rates that emerges from that matrix population model. And what you can see is that to the bottom, species are not doing too well demographically. The population growth rates, this is log lambda, by the way, for those who are familiar with this, is negative. So populations are declining through time. And species at the top in red are actually growing. But we can also predict some short-term responses to perturbances, such as proxies to demographic resilience. What you can see here is species that range from having no resilience whatsoever in white to species that have got a high demographic resilience in dark red, measured as per the damping ratio of the matrix population model. And if you remember how to explain the variation along this two-dimensional landscape, you remember that species at the bottom have the ability to shrink, and species to the left have got the ability to grow fast. That's exactly where the dark red, highly resilient populations are found. So two ways for the plant kingdom to overcome perturbations. One, you decrease in size, most likely in hopes to be able to regain your growth and to continue reproducing after the disturbance is long past. Or the other way is to live very short and to have a high turnover. Let me take you to the fynbos in South Africa. I don't know if there will be an equivalent ecosystem in Brazil. I apologize, maybe you can tell me later. In South Africa, in the fynbos, which is a fire-prone region, two life history strategies coexist: seeders and resprouters. Well, these are your seeders. These are species that reproduce a lot and they die young. And these are your resprouters, species that when things get ugly, they cut down, but then they regrow. And we can explain all of that just with two properties: how long you live and how often do you reproduce. Likewise, we can also link to other ecological disciplines such as functional traits. This is an area that I've been exploring together with Nuri and other colleagues in more recent publications. But here, what we can tell is that by interfacing COMPADRE and TRI, the TRI database for functional traits, we can predict a really important property used all the time in functional trait studies, and that's the specific leaf area. The specific leaf area is the ratio between the area of a leaf and its dry biomass. So you've got a species that has got a low specific leaf area, such as this one, and a species that has got a high specific leaf area, such as this one. This is an expensive leaf to create. That is a cheap leaf because it's thin. What you can see here is that as you walk from fast creatures to slow creatures, the SLA increases. You can also see, however, that there's some exceptions up here. So species that are long-lived and that reproduce much don't create expensive leaves. And the way in which I contextualize that is that there may be a linkage between investments in beta rates, the effects on the physiology of the organism, and a linkage to its conservation biology status. What I did in this paper is I linked the species taxonomy to its current conservation status. That goes all the way from least concern or data deficient down to critically endangered. And on top of that, when the authors in the publications that I use identified the population as an invasive, I also added that new category. What you can see here is almost a perfect gradient going from critically endangered to endangered to vulnerable, not threatened, least concern, data deficient, maybe all over the place. But up here, corresponding to the species that have got cheap leaves, you are dealing with invasives. Yet another way in which demography can help to address the question, what makes a species a ferocious invader? For those of you who may be interested in animals, of course, we have got this great resource, the open access COMADRE Animal Matrix Database. One of the postdocs that I co-supervise, Kevin Healy, now a lecturer in Ireland, and some colleagues and I looked at this same question using animal demographic data sets. And without really getting into the little details about what these Greek symbols and Latin symbols mean, we were able to explain about 70% of the variation in the animal kingdom using COMADRE, not by using just one axis, but by using two. So again, the first axis, you've got species that live fast, okay, like some chicken, species that live really slow, like some corals, some tortoises. And on the y-axis, you have species that schedule the reproductive allocations differently through time. Now, I want to call your attention to the fact that this axis is reversed from the one that I showed in the plant kingdom. So in the plant kingdom, we had iteroparous species that reproduce multiple times at the top. And here we've got semelparous species at the top. But it's a PCA, you can rotate the axis, it's all the same interpretation, okay? And obviously, as humans, or animals, you may be wondering, where are we? Well, you're there. So these are different human modern populations around the globe. And what you can tell here as a take-home message is that, yes, we tend to be long-lived, but we don't come close to many other species, okay? And we're pretty semelparous, But there have been 1, 2, 3, 4, 5, 6 different populations from Japan to Norway. What you can find here is that the schedule of reproduction is quite plastic. And this is also a question that we're currently examining both in the plant kingdom and in the animal kingdom beyond humans. Now, an area of expertise that we've been developing in the last 5 years has to do with a big gap of knowledge for demography, and that has to do with aquatic ecosystems. Historically, a lot of demographic data and a lot of life history theory has focused only on terrestrial organisms. And yet 2/3 of the planet is not terrestrial, it's aquatic, both marine and freshwater. So with the previous postdoc, Paul Capdevila in my group, we wanted to ask the question, do marine species and freshwater species, aquatic species in blue, behave differently from terrestrial species? And as you can see here, again, fast low continuum, an axis that tells you how you schedule in your reproductive events, okay? They explain together about 70% of the variation. Here, you have got both animals and plants, by the way. What you can see is that for the reproductive strategies axis, there's no difference. But that in the fast low continuum, aquatic species tend to be shorter lived than terrestrial species. And this may have great implications for the conservation of those species. Namely, we know that the generation time of a species, as is currently being used in some assessments by the Red List IUCN, is used as a proxy to the risk of extinction, the risk of endangerment. So a follow-up question that we're currently looking at is how species of different realms react in the resilience components differently depending on the kinds of disturbances and human actions. One thing that you might have realized is that there seems to be areas in that PCA that are not populated. The PCA doesn't show, as you would predict, perhaps as a shotgun, a perfectly spherical little mess. Instead, there are some corners of this two-dimensional landscape that are not populated by data. What I would argue is that there are some combinations of life history traits, life history strategies that are not viable, and that there are others that we haven't sampled yet. So here you have got species that are both short-lived and they don't reproduce much. You have to allocate onto some successful vita rate in order to maintain either yourself through survival, for which you need to live on, or the next generation through reproduction. So you have to reproduce. Species, virtual species that will be sitting in this corner would be unsuccessful at recruiting, and therefore they will have been wiped out by natural selection. Species that would sit here, I can actually think of a few, and maybe in Brazil, you guys can give me some further examples. I'll be very keen to explore that together, by the way. Here you have species that don't reproduce much. They're semmelbergs. They have got the ability to decrease in size, and they live very long. Examples from perhaps Africa include the Welwitschia mirabilis, Welwitschia plant, which is a beautiful plant, by the way. If you haven't looked it up, I encourage you to. The main take-home message from this, however, is that there are, in fact, two axes of variation for life history traits, fast low continuum, and one that divides species according to a continuum of investments on the timing of reproduction. Species that just reproduce once are sitting at the bottom, like salmon. Species that reproduce a lot of times, like some orchids, are at the very top. And in between, you've got that gradient from less to more. But we also have some suspicion that this space could be populated, as I just mentioned. And this is also inspired by the fact that chemists keep on finding inorganic elements. The last one happened not that long ago, actually, just a little bit over a decade ago, and it was the tennescin. And more inorganic species keep on being discovered the same way that ecologists and taxonomists and botanists and zoologists keep on finding other species across the tree of life. So we need to better understand the demography of species across the tree of life to really have a good grasp on what's possible and what's not. And I would argue that this question, what's possible and what's not, is a fundamental one to address, because without knowing where the extremes of life history strategies are, we cannot really tell how biodiversity as a whole will be compromised by climate change. There are two things that are for sure, and that is that all IPCC climatic models project an increase in more harsh conditions in mean of temperature and decreases in precipitation. But we also know that the temporal autocorrelation of those events is changing. And I don't need to convince this audience. You are very well versed in this topic. So I'll just skip through this series of highly influential papers to tell you why we need to go places. This is a paper led by a previous PhD student of mine, Aria Panev, now a Marie Curie Fellow in Spain. What we wanted to do here is to understand whether the vertebrate demographic diversity for which we have data is actually represented in the ecoregions where climate change is expected to hit the hardest. What you can see here at the bottom is an index of ecoregion vulnerability going from low to very high in red colors. Incidentally, you can see regions in Brazil that are expected to be highly vulnerable to climate change. Okay. And then what you can see as well is studies that we did in this review with thousands and thousands of vertebrates, okay, for which we evaluated whether climate change effects had looked at the interaction between the demographic rates, these vital rates that I explained before, and the climate, and whether these were negative, neutral, or positive. That is, can we predict in a unidirectional way how climate change is going to affect the building blocks of demographic performance, survival, growth, and reproduction? And are those publications, that fieldwork, is it coming from areas where we need to know the most? As you can see, a lot of the publications are coming from North America and Europe, where in fact, a lot of the ecoregions are not really doing too bad according to climatic projections. Very little data coming from mammals in Latin America that we are aware of as per this literature review, however. Now in the following graph is quite busy, so I'm going to walk you through it step by step. What we did here is for every study where we found an effect of interactions between the climates in the study and the stage-specific demographic rates of survival, growth, and reproduction, we evaluated whether there was an intensity that was nonlinear, positive, neutral, or negative. A nonlinear relationship would imply that, for instance, as you increase temperature, perhaps investments on survival first go up, but then go down. A linear would imply that it just goes in one direction in a linear manner. What you can see here is an example for a meerkat. I think that these data are coming from Arpad Osgul's lab in the field took place in Colorado. What you can see here is that the three vital rates are nonlinear. Likewise, you can see for many other mammal species, the results are all over the shop. The effects of climate change or climate as experimental study in these publications on different demographic rates is very much species specific. But now we do know a way in which we can classify how the species may fall according to different life history traits. So we may be able to do better than just looking at that big picture. Now let me take you back to climate change. As I said before, things are going to get in mean values more harsh, but they're also going to get more harsh in terms of the unpredictability. So what we're more likely to see in the future going forward is, instead of these extreme events, we're more likely to see changes in the temporal autocorrelation. And there's been some papers in demography that have looked at how different populations in very small species numbers might react in their performance to good years and bad years. And they've done that typically in IID environments. That is, the chance that you stay in a good year or you go to a bad year the next year is the same as that you stay in a bad year or you move forward to a good year. But again, what we expect in climate change is an increase in the positive temporal autocorrelation. So what we did in a study, again, with Maria and with Arpad, is we evaluated this question using demographic data from COMPADRE and COMADRE and using mathematical simulations. So effectively, what we asked the question for was, what happens if instead of going from good to bad to good to bad, you increase the temporal autocorrelation in a positive manner? So you're in a bad, you're going to stay there for a long time. Before you switch to a good here, you're going to stay there for a potentially long time, and then you're going to switch back and forth in a positively autocorrelated manner. What we did is for 454 animal and plant species, we structure them again according to this periodic table of the elements from chemistry, but now using life history traits. What you find is the PCA 134%, again, fast to the left, slow to the right. The second axis is explaining 33%, semioporous to the bottom, heteroporous, reproduces many times, at the top. And what we put in there is, again, the same life history traits, namely, can you reproduce a lot or not, and how long do you live? What I'm showing you here is one species population as represented per each dot. And you can see there's some examples of plants, you can see some insects, you can see monkeys, corals, trees, etc., etc. The important thing here, though, is to pay attention to the radius, the size of that dot. What I'm plotting here, what we did is we calculated the sensitivity of the stochastic population growth rate for each population with respect to changes in the environment in a way that would be positively autocorrelated. Okay? And so what I just said, which is a whole mouthful, is the same thing as saying we calculated the vulnerability of these populations, the natural populations, to climate change as predicted by the IPCC scenario. What you can see is that contrary to predictions of life history theory using IID environments, we find that short-lived species are going to be more compromised by climate change. So this is, when you compare that to the previous research, where we find that understanding generalities from vital rate responses to climate change is really difficult, this really gives you a really strong take-home message, and that is short-lived, not doing well into the future. We've expanded some of this research using some more advanced statistical techniques to evaluate how extreme events of precipitation and temperature, defined as years where the precipitation and temperature is farther than 2 standard deviations from the long-term mean. Okay, so we're interested in looking at how these extreme events may react or affect the population growth rates. And we're also interested in looking at how life history strategies may be good predictors to predicting positive or negative responses to those effects. This is work that is currently in review by one of the postdocs that I co-advise, Aldo Compagnoni, in collaboration with Tiffany Knight at IDIF as part of a working group that I'm leading there. And what Aldo found out is that the total effect of extreme events in precipitation and the total effect of extreme events in temperature drastically shapes the demographic responses of both long-lived and short-lived species of plants. What you can find here is in the x-axis, the generation time. Again, on log scale, species close to 0 are short-lived species. Species with values of 4 will be quite long-lived, okay? What you can see here is that the absolute effect that extreme events of precipitation have on the population growth rates of these species is really significantly predicted by precipitation, the interaction between precipitation and generation time, less so on temperature. And the degree of parity, a proxy to the second axis of the PCA that I showed you before, is not a good predictor to demographic responses based on extreme events of precipitation and temperature for plants. So this is really important because we, with this information, we can go back to the IUCN and start to encourage them to use for the first time generation time, just as they do in the animal kingdom, consistently across the plant kingdom and not other life history traits for measurements of potential vulnerability. So what this means is that we're potentially looking at an erosion going forwards in this 2-dimensional volume, whereby fast-living species might be compromised. We may be dealing with species that we nowadays have. If you go around the road and you'll see rudels, but if climate change kicks in ways that it's definitely affecting plants and animals, we may be depending or moving on to depending on other more long-lived species. And more recently, my group is now looking at other axes of variation. Together, these two previous axes, the fast low and the IT-Roper similarity axis, explain about 70%. But we, of course, want to explain as much as possible within reason. We have evidence that the investments on reproduction and how the change in reproduction occurs with age can be completely decoupled from actuarial senescence or so-called survival senescence. So that's a third axis that is orthogonal. Also in collaboration with colleagues from Brazil, Gabriel Santos, and with Maya Kajen, we are looking at other axes that has to do with investments on demographic buffering versus stability. So I'll be happy to discuss that with you guys if you would like. But just to show you the decoupling between actuarial senescence, that's the red line, and reproductive senescence, that's the black line. What you find here is a paper that is currently in review again by one of my PhD students, Mark, where we classify the age-specific fitness components of different plants and different animals. To the left, you've got species where the survivorship curve in red declines, as you might expect, very quickly. And to the right, you've got ones where it declines less quickly. In black, you've got age-specific reproduction. And what you can see is that if you decline for one very quickly, you don't need to decline for the other fitness component very quickly as well. In fact, investments on age-specific survival and reproductive trajectories can be, and they are indeed, quite decoupled. We've evaluated this for hundreds and hundreds of plant and animal species, and the same image emerges, that there is a novel orthogonal axis that has to do with independence on reproductive senescence. Together, these three axes are allowing us to use demographic data and life history theory to understand anatomical constraints, project demographic performance, evaluate whether senescence or aging is universal, predict IUCN status, and many other questions, some of which you can find publications highlighted there, and I invite you to take a look at them if you are interested in them. I would like to leave you with a message that we now have got a framework with which to classify life across the tree of life. We can understand the relative impacts of phylogenetic constraints and environmental filtering, and also make really invaluable projections for climate change. I'd like to end on three notes, if I may. The first one is to thank the working group that I lead, called S Apropos in IDIF, involving researchers from all over the globe. I'd also like to thank my various funding agencies. I'd like to make a call if any of you, particularly PhD students and potential postdocs, are interested in some of the questions that I described today. Get in contact with me. I am particularly interested in bringing more diversity to ecology and evolution. And I think that the talents from Brazil could definitely help in the current crisis in biodiversity. And on that note, I would like to finish by thanking the audience, and I'll be happy to discuss and address any questions.
Thank you very much, Roberto.
Thank you.
Se alguém tiver pergunta, dentro do Zoom, quem quiser falar pode abrir o microfone diretamente, e quem não pode escrever lá no chat a pergunta em português ou em inglês. E lá no YouTube as pessoas também podem deixar pergunta que a gente vai passar aqui no Zoom. So when we are waiting, I want to ask you about the IPM, this database that you are developing, right?
Yep.
That you were looking at the beginning. And which are the differences between COMPADRE, COMADRE, and the new ones?
So they're pretty much the same thing in terms of how the data are archived in the field. They are accommodated in a slightly different and some would argue more powerful format. Okay, so you can think of a population in terms of how different individuals in that population are. You can think about that population being bacteria. You can think about that population being us humans. You can think about that population being a population of lions, right? And what you hope to achieve in building a useful demographic model is to classify individuals in that population in a way that you accommodate for differences that affect survival and reproduction, okay? So for us humans, for instance, age is a really good proxy, okay? Another proxy arguably could be perhaps the nationality, the socioeconomical background. How many cigarettes do you smoke? How many glasses of wine, etc., etc. All of the traits, or most of the traits that I just told you, are discrete. Age is discrete in a discrete time step model, like a matrix population model. But in reality, a lot of the traits that are really useful to understand survival and reproduction are not discrete. They are continuous. So things like size, things like volume, that gives you competitive advantage, things like height, things like the color of your eye, if that's what ultimately allows you to mate successfully. Colors of eyes don't come in just 3 flavors, blue, brown, and green. They're full spectrum, full continuum. So an IPM, also known as an integral prediction model, allows you to use the power of integrals to integrate, sorry for the redundancy, across the continuum traits that helps you predict survival and reproduction. And what we are doing with these databases is we're, together with the database itself, we're developing an R package called R-Padrino that helps the users re-parameterize the parameters from each IBM for publications and to obtain their own values as well. What's interesting about IBMs, and is really exciting, is that the rate of accumulations of literature using IBMs at the moment is greater than the rate of accumulation of matrix population models. So in other words, Padrino and Madrina are smaller because the first IBM paper was first published in 2000, okay? But they're growing faster than Compadre and Comadre together. So stay tuned for that. Hopefully we'll be able to release it next year, pandemic allowing for that. Have I lost Nuria? I am afraid I might have lost Nuria.
Now I think we can unmute because something happened with the and Roberto, in the same following Nuria question, I'd like to ask you how is it's because I'm teaching now IPM, then after I learn with you the IPM, and I'm teaching the post-graduation program, and I have some students here from my class, and I'd like you to, maybe you can tell us, I think we more or less explained it for us, but how big is the Padrino and Madrina? Is it the same size, or it's far from Compadre and Comadre?
So we are at the moment is smaller because, as I mentioned before, the first IBM paper was published back in 2000, right? So it's only been 20 years. Compare that to the first paper that used matrix algebra to describe a population, taking you back to Leslie's work back in 1945. So a long time has elapsed since that. In Compadre, we now have got close to 1,000 plant species. In COMADRE, we have got over 600 and more are being digitized. As we're speaking, COMPADRE is no longer just based where I am. COMPADRE and COMADRE are a consortium of 10 different universities and research institutes like the Max Planck and IDEF. So it's growing quite fast, but in answering your question, Padrino and Madrina currently have got, that have been digitized 300 plant and animal species, okay? But that are awaiting digitalization by my team, hundreds. So we are receiving an average of a paper containing IBMs that I find on the literature using Google Scholar and ISI and Scopus, a paper every week. Now, how many weeks per year and how many years since 2020? Sorry, since 2000. Obviously, it's not going linearly. It's growing exponentially, but it's a really nice, beautiful challenge to have because we're having to develop very creative ways of funding this work. A lot of funding agencies here in the UK and in previous countries where I've been based don't allow you to do this type of work, database work. They want you to do the research, but guess what? Without this type of work, you cannot do the research, at least if you want to use a demographic comparative lens, right? So we're doing that through a lot of volunteering, and we're doing that through some small investments from pockets sometimes even. But the databases are going forward nonetheless. There's, you may be able to find, so the database itself will be released hopefully sometime next year, but you may be able to find this GitHub that serves as the intro, really plain slate intro slide to Padrino and Madrina. Okay, this is the GitHub of Sam Levin, one of the PhD students in my group that I co-advise with Tiffany Knights in IDEF, and he's leading this effort. And if you go to his GitHub, you'll also be able to find the R package, which, by the way, is very much still on beta mode. But we encourage you to use it. If you're teaching your students, we would love to have your students as our guinea pigs, if you would allow me. You may be wondering, Rita, yeah, but what's the difference between this new package and IPM Pack, right? Which is another package that we developed with Jess Metcalf. Well, I think that the IPM Pack was a really successful intro level or medium level R package. But for more advanced analysis that require really understanding the guts of an IPM, it had that somewhat limited value. What we're trying to do with our Padrino is create full flexibility. But anyways, this effort is led by Sam, one of my PhD students with Tiffany Knight. So I encourage you to obviously get in touch with me if you want, but give credit to Sam. If you like what you're seeing, let Sam know.
Thank you, Rob. And I think we have two more questions here. Maybe Marquinhos, could you turn on? No, just a moment.
No worries.
There is something happened with Zoom here. I don't know what's. I don't know what Nuria did. Now we have Mark.
Okay. Okay. Can you hear me?
Yes, very well. Hi, Marcus.
Okay.
How are you?
Hi, Rob. Really nice having you listen to your talk. My question here that's just wrote in the chat. It's about, it seems to me there's a contradiction between when things get ugly, the predictions on life history. What would happen? Who would be favored when things get ugly? And it seemed to me that would be the fast or semmelperous species. But actually, when climate change happens, they are actually doing worse. Is that it? Or and if that is so, how do you explain that?
So I think that obviously to reduce this message to just a very general statement like that is quite daring. But given the time allocation of 1 hour, that's as much as I can say. But I'm happy. And I thank you for giving me the opportunity to elaborate on that. Of course, it's not it. What we're currently doing is we are decomposing different perturbances, right? So humans and the environment can affect biodiversity in many ways. and each way could target a different life history trait differently. I'll give you an example. If you are a large organism, but you get hit by a drought, if you have got storage in you, you might not be hit as hard as a small organism. Okay? So droughts might affect size differently. However, if you're big, you're more obvious, you're more visible. So you might be harvested, you might be hunted with a higher likelihood. So what we are doing at the moment is we're developing, with a master student of mine named Thomas Marin, we're developing an application of a resilience framework also done in my lab, where we are decomposing rates of extinction based on different human activities. I don't know if you guys are familiar with the human footprint database. So the human footprint database, for those of you who may not be familiar with it, I highly recommend you take a look at it, by the way, it's great, is a terrestrial worldwide map broken down to 1 square kilometer of different activities, human activities, into 7 different layers. So things like crops, urbanization, light pollution, proximity to a road, etc., etc. And what it really is allowing us to do is to break down this question that you're asking me. That's very much work under development, but we are finding very striking differences depending on the kind of disturbance shaping the extinction of one species or the other. One thing is still remaining true though, and that is that IID environments, truly stochastic environments, don't happen. We've taken an IID approach in the past, the field has taken an IID approach in the past out of pure mathematical convenience, because it's way easier to parameterize and to project a model where things don't have complications, more reality.
Marina, corre.
The temporal autocorrelation. But I think that being able to break it down by layers really will hopefully add more flavor, more findings.
Tô saindo aqui do Tintim.
Alguém estava com o microfone aberto. I'm sorry, Roberto, my internet was like crashed and
I guess I don't worry about it. Rita took over the show went on, you're fine.
I'm sorry, thank you. And I don't know if there were questions in the chat, but Andre,
Oh, thank you, Rob, for the answer. Yeah, it was very.
Thank you for your question. Excellent question. Yes. Yeah, it's not that simple, really. I mean, what some papers are describing is that bigger species are more likely to go extinct, and that has to do with things like, if you're bigger, you're more obvious. You're a more obvious target, depending on the activity, right? But with size also comes an increase in competitive ability, for instance. So being big is not always bad.
Actually, I came across a recent paper on birds, comparing different species of birds and how their populations, or they persist under stress or effects or changes in climate changes or changes in the environment, disturbance in general. Yep. And actually the large-sized ones, they do better than the small ones, looking at just size, body size.
Yep. Of course, there's a really interesting aspect there when it comes to understanding responses of birds to climate change and the environment in general, and that is that for most of birds, not for all of them, but for a lot of birds, they literally have got a flight escape ability that many other organisms, not just plants, right? All sessile organisms across the tree of life, and all organisms that cannot migrate long distances, they're stuck where they live. So from that very perspective, you would expect that birds will be a bit of an oddity, right? Birds and big mammals, by the way, because they can also migrate long distances, and therefore decouple a little bit the environmental change from their demographic responses. But then again, also that comes at a great energetic cost. So the story is really interesting, deserves more attention.
Can I have a kind of following up question? First, thank you, Rob, to be here, and really nice to have you here, and thank you for this interesting talk.
Thank you.
And I was also struck by this prediction that short-lived species would be more vulnerable to climate change. But yeah, I think it's logical the way climate change acts with these longer periods of like bad conditions. But if we think that many of these direct human impacts actually favoring these species, short-lived species, because of the increasing in disturbance regimes. You kind of replied this already, but I was thinking if in a scenario when these two things are coming together and interacting, so you have from one side human increasing disturbance and you have favoring short-lived species, yep. And on the other side you have climate change happening, and then you have those species being yep. displaced, and what the scenario is kind of catastrophic.
Yep. So I think that would be
interactions also.
Yeah, and interactions is a really difficult one to, I think, for the time being, bring into this approach, because the data are big, but they're not as big as they ought to be in order to test these questions, right? You very quickly can run out of power, even if you're dealing with hundreds of species. But anyways, I'll deal with the first question though, and that is that, yeah, absolutely. What I think would be really useful ultimately is to be able to provide region-specific recommendations. Okay, so if you are, I don't know, 20K, I'm gonna make it up. I don't know exactly what the south of Rio de Janeiro looks like. I've never had the pleasure of visiting your beautiful country, by the way. But if 20K south from Rio, there's a specific kind of disturbance that overdrives or dominates the biodiversity, we could perhaps fine-tune this framework with more data to be able to make more predictions. What life history traits and strategies are you likely to lose? And there, to be able to work with land managers to put remediation strategies and restoration strategies. Go to Norway, same. The climate's going to be very different. Most likely the kinds of human disturbances are going to be very different. We already know that Norway is projected to be less vulnerable than Brazil from the map that I showed you before. Predictions will probably change. So it's, as everything in ecology and evolution is got to be a tailor-made prediction and a tailor-made solution probably.
And I guess then it will depend a lot of the strength of each of these impacts then, and the combination of them. Right?
And one thing that I think is really promising is to keep on using theoretical approaches and modeling all of this data, right? So one of the things that we're currently looking at is a bit more complication, a bit more richness on this environmental change and autocorrelation. We know that when it comes to disturbances, be them indirect human-led like climate change, or direct human-led like habitat degradation and fragmentation, we know that the effects are not linear. Okay, we know that disturbances can have different non-scaling effects. So if you have a huge one-time disturbance, the biodiversity responses will be most likely different than if you say half that big one event and put it year after year in a consecutive manner. So what we're really interested in now, a couple of grants in review at the moment, hopefully one will be at least funded, is evaluating the sensitivity of different demographic properties of these models to different moments of disturbances. So the intensity is one, the frequency is another one, the stage specificity, going back to the point made by Marcus, right, is another one. Interactions between different disturbances as well. What I'm proposing to do in that grant is actually looking at the 6 different types of disturbances. And we can do all that through the magic of big data theory and a bit of creativity and a lot of R programming.
Thanks.
Thank you, Andre.
And now we have more 2 questions here, Rob. It's from Mariana and Vinicius, if you can.
Hi, Rita. I can.
Hi.
Mariana.
Thank you.
Hi, Mariana.
Thank you for the talk. Great talk.
Thank you.
You have these two huge database, COMADRE and COMPADRE, and you have some matrix population models for like tons of animals and plants. And you analyze all this data and you give us like this general pattern. But I was wondering if you have like an effort to understand how, like in this strategy, the life history strategy, how the animals or plants, how each life history trait respond to like climate change. If you know, or there's like an effort to understand how these different species, survival or reproduction, how they respond to these changes in the environment.
That's an excellent question, Mariana. Thank you for that question. So we are doing a little bit of work on that, but we find it a bit more promising to look at the full syndrome. Okay, Mariana, if I may ask you, how tall are you? Give me a number. You may be like 1.6 or 1.7 meters.
Sorry.
How tall are you?
Oh, I can tell in English.
Just give me. Let's say I have no idea how tall you are, but let's say that you're 1.6 or 1.7 meters. Okay, let's just go.
Yeah, meters. Yeah.
Okay, that number doesn't define who you are, really. That's it. What defines your survival, what defines your reproduction is probably how old you are, how tall you are, how much you weigh, how healthy you are. Again, do you smoke or not? So it's the full syndrome. It's a full coordination of different traits that defines who you are, and most likely defines how you react to having an annoying neighbor or living close to a road. Now, let me take it to this framework. There's been a vast amount of work that has looked at longevity, particularly. Okay. This work is coming from aging research. As you probably know, humans are obsessed with wanting to live longer, but not just wanting to live longer. We want to expand health span. So instead of lifespan, we want to expand the good years of life, the health span. So from that perspective, there's been a lot of genetic research and a lot of socio-economical research looking at how do humans get to live longer? Why are babies born in Japan this year, on average, going to reach a mean-life expectancy of 100 years, which, by the way, is true. Okay, mean-life expectancy. You're born in Japan this year, you get to be a centenary. Whereas if you go to Spain, where I'm from, your life expectancy is long, but not nearly as much. Okay, so there's a genetic component, there's a socio-economical component, there's a nutritional component. We know that certain species, when they don't acquire a lot of resources, they grow fast. Sorry, they grow slow and they live long. So this is all linked to the theory. This is all linked to the gigs of the life, right? This is all linked to the theory of caloric restriction. So there's been a lot of vast amount of research evaluating particular longevity, perhaps not so much moments of reproduction, to be honest. And I think that that's an open area, really. So if you guys, Mariana, yourself, or if anybody in the audience is interested in that, I think that the field is mostly wide open, yet waiting exploration. In my group, we find it really interesting to look at the whole combination, because that's ultimately what, from a demographic perspective, defines the full range of possibilities at the organismal and the population level.
Okay, but can I just add a little? Because when you have like you have to do some conservation program, and you are focusing on one species, and you want to know like which like moment in the life cycle the species is most sensitive to some disturbance.
Yep.
And is possible to do like this big analysis, focus on these kind of questions, like saying for some mammals, the most sensitive
of
the life cycle is like juvenile survival, like something like this.
Yep. It is definitely possible, and it's only possible to do that kind of analysis once you have got a good understanding of the full life cycle. So we're back to the need to understand the full syndrome. Okay, so you can only understand, you can only reach the conclusion that, for instance, in a specific species of mammal, adult survival is the most sensitive stage and demographic rate to population growth rates if you have the full life cycle. Otherwise, the analysis doesn't work. You need data across the entire life cycle, from offspring all the way to very old adults, and then the loop back through reproduction.
Okay, thank you.
Thank you.
I have another question. Can I ask?
It's, may I ask? Oh,
No, Vinícius, vai lá. Vai lá,
okay.
Vinícius.
Okay. Quite fast. I'm not sure if I should start with my question, but because my question is about open data, maybe if you want to make a question before about the talk itself, maybe it's better to you, Marquinhos.
Okay, my question is about phylogenetic signal. So, you you mentioned it is really weak in plants. I just know if it's, you can say the same about animals.
Excellent question there. Yeah, so the answer is no. The answer is, for animals, we find the flip side of the story. In animals, we find the phylogenetic signal to be much stronger than in animals. In plants. So Pagels lambda, which assumes Darwinian, Brownian model of evolution. And there's something wrong with that assumption, as you probably know. We can discuss about how to improve on that. But that model, assuming a Brownian motion kind of evolution, finds that for animals, Pagels lambda is between 0.7 and 0.8, which is really high, so long as comparative analysis go. So what this means is that if you're a mammal, allow me to now bridge back to what Mariana was asking. If you're a mammal, your demography is likely to be like that of any other mammal. Okay? If you are a bird, your demography is likely to be like that of any other bird and more different than that of any other mammal. Okay? Now, let's think about birds for a second. A bird is a bird is a bird. And I'm sure that I just insulted a lot of ornithologists in this group, and I apologize for that. But what I mean by that is that while birds can differ in maybe 2 or 3 orders of magnitude in biomass, okay, plants differ in 10. Okay, and not only do they differ in 10, within a specific taxonomic family in plants, you can find herbs, you can find shrubs, and you can find trees. In Fagaceae, for instance, okay? You've got all forms, or not all forms, but a lot of different taxonomic forms, growth forms, within a specific taxon, and that is a unique family. And if you believe, as I think it makes sense, and I have got evidence for, that your architecture fundamentally shapes your demography, if your architecture is not conserved at the family level, your demography shouldn't be either. Now, mammals, there's a lot of oddities within the mammal clade as well, obviously, but mammals are more or less the same. Again, I'm sorry if you're doing mammal ecology and I just insulted. I hope you understand I'm trying to oversimplify this question, right? Plants are a lot more modular, they're a lot more plastic on average than animals. There are big exceptions though in the animal kingdom, and those exceptions look really similar to the plant kingdom, namely corals and sponges. Corals and sponges vary a lot in their demographic signature.
Great. There's just a comment. There's a comment of paper of Mariana and I comparing marsupials and placentals, the two main lineages, and they're quite different in the fast.
You would expect massive differences in terms of maternal and paternal care, for instance, right? In a main effector. I would love to, if you could remind me later, take a look at that paper.
Okay, great.
Well, may I ask my question about open data? Okay, good. And first, thank you for the talk. It was very nice and very instructive talk.
Thank you.
And first, I should introduce myself. I'm Vinícius Farjala. I'm a professor in the same department of Rita and Marcos Vinícius, who make this talk, this questions for you before. And well, there is a lot of pressure about opening data, mainly in the tropics, and you pointed out this topic in your talk. And what I'd like to know if there is some discussion about how to acknowledge the data providers.
Yeah,
right.
Absolutely.
Well,
Absolutely.
This is something that I have been thinking about, how to better evaluate the work of these data providers.
Yep. This is such a fundamental question right now, as ecology is aiming to become bigger, more extensive, and more predictive. And without big data, becoming predictive is going to be really, really challenging, I think, because you will end up being able to make predictions about 1 square centimeter in your backyard and not being able to upscale to other regions, right? So I think that the issue of giving credit where credit is due is fundamental to not just ecology, it's fundamental to science, and it's fundamental to collegiality. So there's different models in terms of how to approach this challenge. I can tell you a bit more about ones in which I'm not involved, but I think I might try to tell you about the ones that I know a bit more, if that's okay. So I, as you know, lead COMPADRE and COMADRE with other colleagues, and I'm also involved in the Open Trades initiative. So let me tell you about the first two, and then I'll tell you about Open Trades. So one of the things that we've been very conscious to do in COMPADRE and in COMADRE, and also in the IPM databases, is to archive the full citation of the papers from which we are digitizing the data. Okay, in the papers that I wrote with my collaborators when we first introduced COMPADRE and then COMADRE, we made a call for the users of COMPADRE and COMADRE to cite the original resources. We made a call not just by saying that, but we also developed the R functionality. So if you go to the COMPADRE GitHub, I can show you a link later. We've developed scripts that once you have got the list of DOIs from your study selection in COMPADRE or COMADRE, so DOI is a digital object identifier, and each paper has a unique one. Okay? So if you give me a DOI, I'll be able to download the full citation. So what we're hoping to do is to make the life of the users easier by facilitating giving credit where credit is due. In quite a few cases, we know that getting access to the data, if the data is open access, is one part of understanding an ecological or evolutionary question, but really interacting with the users is priceless. You see, Vinicius, I can give you data that I collected for my PhD. I've got 16 years of data. And in fact, those data are open access. So you wouldn't even need to contact me. They're open access in ecological archives. You go there, you might be able to navigate it if I've done a good enough job at archiving the data, but you will never be able to understand the idiosyncrasies of the field sites. Because for that, you need to be there, right? So if you find something really quirky or something really interesting, you might want to contact the original author and establish a relationship whereby things perhaps might even result in a collaboration. And I know of quite a few testimonies where that has been the case between users of COMPADRE and data contributors of COMPADRE and COMADRE as well. So we're hoping that really that's a model. I know that a lot of journals, like Journal of Ecology, where I am an associate editor, no longer put a cap on citation. So you've got your main citations in the main prints of the paper, but you can add as many citations as you want in the online appendix. And those, importantly, get archived in Google Scholar, in whatever have you. So you get citation impact, which is super important, I think. Now, another thing that we do in Open Trades, so Open Trade is a platform of databases. So if you go to opentrades.org, you'll be able to see what we're doing. Opentrade.org is a platform where people who are either actively using big data or people who are contributing with big data, like my group, for instance, come together. We think about what are the good practices in open science. We've developed a paper with some recommendations that was published this year in January, led by Rachel Gallagher in Nature, Ecology, and Evolution. And we've got a really active debate about how can we really get past how awkward this conversation can be and make it beneficial for the data provider, for the data user, and for the discipline.
Okay, thank you. Very nice. It's a very Very nice explanation. Yeah, that's something that I have been discussing here in Brazil, and I feel that most of the novelty, it comes from Europe, from US,
Yeah.
from Canada, and we just like receive this news, and we never know quite well what's going on. It's very nice to hear from you.
Yeah, and I think you know, I am aware of, I'm going to say something that perhaps is politically incorrect, but I will still say it. I am aware of some international frictions that have emerged between certain countries in terms of going to a field site, collecting data, going back to the original country, and there not be a linkage between the country where you did your fieldwork and the country where you're actually writing the paper. You guys probably already know what I'm talking about. I'm just not going to name countries, okay?
Sure.
I think that that model was bound for failure. Just like a model where you're using people's data but not giving data credits is also bound to fail.
Well, yeah, that's also my point of view. I agree with this open data. I pretty much agree with this one, and I advocated for open data.
Mm-hmm.
But on the other hand, I have these concerns about how people use this data, or to, because it's very common for at least in science. Science, it's quite good in evaluating high impact factors and the first authors or people that are publishing these high impact factors.
Yeah.
But on the other hand, science is quite bad in evaluating the people that provide the data and that go to the fields, and they are
really
more or less equally important in the whole system, in the whole process. Right?
I would definitely agree with you. I mean, if oh, sorry, go ahead.
But yeah, that's very good to hear that you, for example, this experience that you told about the Journal of Ecology. It's very nice. Yeah, that's you open the number, you let open the number of reference, and people can add reference from data papers and the papers that they use it, or information.
Exactly.
That they collect.
And what we're doing now, I mean, my experience with Compadre and Comadre has obviously been really positive. I've not had a single, and I promise you this, a single negative experience in trying to lead that or having nasty interactions with users or data providers, okay? One of the things that we're currently working on with the support of the NSF in the US is developing a new version of Compadre where users are able to, sorry, not users, data contributors are able to go to the database and deposit their information. So the way that Compadre and Comadre works very much up until today, and probably tomorrow too, is by typing. A lot of digitalization that my team and other teams around the globe are doing. As I said before, most funding agencies don't give you money for that, okay?
Yeah.
So that's happened mostly out of love for science, and because we also use the data to address questions as I presented today, right? What we're working on with database architects is a system whereby you guys or anybody that has got demographic data can go to the database, archive the demographic information. It wouldn't be released immediately, by the way. You can put it in an embargo. You can say, you know, I'm going to give you this data, but I don't want to see it going live until 2024, and that's fine, okay? It wouldn't go live immediately either because we still need to curate it. Okay, but what that does is the moment that you archive this information, you get a DOI. You can take that DOI and put it in the online appendix of your submission. It pretty much becomes a triad for demography.
Well,
and my experience with authors who have contributed data has always been so positive. Really, authors are very keen to just give you data, and a lot of them, they don't even ask questions, you know, they just give you the data, not questions asked, and it's been really inspirational to work in this field for that reason.
Even for tropical authors?
The only authors where getting data is a bit trickier is, understandably, species that are critically endangered.
Okay.
And species that are highly charismatic. Okay, so think about the tree of life. Think of a species that might be highly charismatic. You're probably not thinking of bacteria. You're probably not thinking of snails. You're probably thinking of tetrapods, okay? And within tetrapods, think about something that is critically endangered. And we probably have had some long conversations, but in the end, people do give you the data.
Oh, good. Good to know. Thank you.
Thank you.
Well, I came back. I was a long time. my internet crashed. Like it was the first time that the internet was so bad. I'm so sorry. And I wanted. So I don't know if someone asked you about what do you think, which are the next steps in your research? Like, because you were talking about this reproduction senescence, the continuum, the new one that you are working on.
Yep.
So I was curious about that.
So there's quite a few areas that I think I'm really excited about, and people in my group and other collaborators are currently working on. Not to blow my own trumpet or yours, I think that the work that you and I did at the interface between demography and functional traits requires further attention. So being able to evaluate what syndrome combination of traits really predicts demography is really useful. From the point of view of a lot of these demographic models require years of tedious data collection before you can say anything meaningful, okay? Now, in 2018, I wrote a paper in functional ecology called something along the lines of the promises of functional ecology and demography. And the point of that is that for functional traits to be functional, they should be able to predict fitness components, survival and reproduction. That is the core of demography, right? So I think that further exploring that is something that I hope to be able to continue working with you. Other areas have to do with where around the globe and under what nutritional cases those species escape from senescence. We're uniquely placed to be able to address those questions in that we have got age-specific trajectories of survival and reproduction, and we have got macroecological approaches to allow us to address those questions. Another area that we're very active on evaluating is in collaboration with two colleagues from Brazil, in fact, Gabriel Santos and his PhD at Zoologia is evaluating temporal responses to climate change. That has to do with whether species can buffer against climate change or whether they just act labile, that is, they just go with the flow. There's another areas that I'm really excited also in lab-based approaches, but they take us away from demography.
Yeah, Maya is actually here. She wrote in the chat that thank you for the fantastic talk, Rob. I can't make questions at the moment because I have a very active baby in my lab, but I'm grateful for the interesting discussion.
So for some reason I cannot see the chat, but that's fine. Hi, Maya.
And I was reading a very recent paper from Laughlin that he was saying that this when we are exploring the trade-offs between the sensitivities and elasticities, he was saying that it is better to use the relative growth rate better because of such trade-offs that can like obscure these interactions. I don't know. What do you think about?
So I think two things. I think very highly of Daniel. I think that he's doing some really interesting science. I think he's leading the way as well at the interface between functional traits and demographic performance. However, I would like to make a plea to stop perhaps looking at long-term dynamics like population growth rates. I'm saying that, I hope you realize, I'm saying that, well, I have presented work where I've used population growth rates, okay? Rather than long-term dynamics, which you can explore from these models, which make a lot of grotesque assumptions, such as density independence, environmental stationarity, etc., etc. I think it makes more sense to look at short-term dynamics. So, and in a paper where you and I are collaborating, what we show, so I guess this is not an answer to you, but to the rest of the audience, right, is that functional traits are better predictors of transient dynamics, short-term dynamics, than they are of long-term dynamics. And that makes complete sense. I mean, as years go by, you lose your predictive power. You also lose the ability to engage successfully with managers. Managers don't want solutions for 10 years. Managers want solutions for tomorrow.
Yeah, that's true. That's true. Thank you. Marcio, do you want to open your microphone?
Yeah. Well, thank you. Thank you, Dr. Salguero, for the great talk. Lots of stuff to think about. And my question goes on this, I think a lot of people asked you about this, with the short-lived species being more prone to negative responses to climate change. But on the other hand, we know from theory of natural selection that short-lived species tend to respond better in terms of evolutionary terms to these effects. So do you have any ideas, or you like to speculate on the long-term ability to respond to climate change as adaptive responses on these short-lived versus long-lived species?
Yep.
Thanks.
So thank you for your question. So all else being equal, so ignore the environment, ignore biomass, ignore everything you know about ecology and evolution. Species that has a faster generation time will be able to adapt faster. Okay. Yeah. Now we know that, and by faster, a shorter generation time is an annual species or something that is really short-lived. However, we know that together with an increase in generation time, biomass increases. So this is kind of like leading me back to the point that we were discussing before with a previous participant. Body mass is a really good predictor, as it is age, of how you respond to the environment, to your fitness component. So the answer is really complicated because you're not keeping the arena constant. As you're growing into a longer generation time, your body mass is scaling up. Your storage is probably scaling up. As I mentioned before, you're becoming probably an easier target to predate, depending on whether you have camouflage or not. But there's trade-offs, physiological trade-offs that are playing fundamentally restrictions that allow species to better accommodate impacts of climate change or not. So, answer to you, and I'm sorry to give you something that perhaps might not be very satisfactory at the moment, is not that easy, right? Short-termness is a good predictor, but it's not everything.
I think we lose Nuri again.
Oh. Yeah.
She was looking so serious.
Yeah.
Yes.
It is true. It never happened before in these talks.
Yeah.
The connection was always very good, very stable. Yeah.
These things
So,
are meant to
Rob,
happen.
yes. So I think we have no more questions here.
Okay, excellent.
And so I would like to thank you so much for your talk. It was wonderful, and thank you to stay here with us almost 2 hours. So thank you very much.
It's been a pleasure. As I said before, I'm very keen on attracting talents from areas that, in my opinion, are not very well represented in European ecology. So if any of you are interested in any of the research that I've presented or anything else that we're doing in my group, do feel free to get in contact.
Okay.
Excuse me, could I make one last question?
Yes, yes.
Yeah.
Yes, go ahead.
Hi, Roberto. Thank you for the talk. It was really amazing. So I am one of the Portela students who are studying demography in Atlantic rainforest. And my question is that most of the demographic studies results are performed in like 3 or 4 years. So, and probably you are losing a lot of information, mainly for like demographic parameters, mainly for the long-lived species, right?
Yep.
So, how could we improve it to get like the maximum and the best information for our data? What do you think?
That is an excellent question. What was your name again?
My name?
Yeah.
Eduardo.
Eduardo, that was an excellent question. So obviously, you might have heard me talking about how many species we have in the databases, right? So I said together we've got 1,500 or something like that between plants and animals, and it keeps on growing. Now, the papers that I presented today don't have that high number. They have 300, 400, 500. You may be asking why. Well, we impose a very rigorous selection criteria onto the species that we use for our analysis. And the selection criteria changes depending on the analysis that we want to perform and also on the questions and the hypotheses that we want to address. Okay? So for instance, if we're looking at temporal autocorrelation and we want to understand how the demographic rates relate to the environment, you don't really do much for 3 years, as you very eloquently put. You probably want something that is more better replicated through time. So for instance, for the paper with Maria, one of the two of them, we looked at species with more than 7 years. Now, obviously, as you become more picky in your selection criteria, you lose more species. There's only a handful of species for which more than 30 years of demographic data exists. They do exist, and we have them, but there's not many. And so it's a matter of quantity versus quality. It's a matter of generality versus specificity. There's a lot of things that you can do with more long-term species. There's a lot of things that you can do with species studies that are better replicated in space as well, but there's less generality. So you have to think very carefully about your questions and make compromises. We all do. When you woke up this morning, you had to make a compromise. Do I shower or not? Right? It's the same thing for animals, and it's the same thing for the rest of animals, and it's the same thing for the research. So in this analysis, depending on the questions, we evaluated, okay, if you're this long-lived, then, and if I've got less than this many years of data, then you're no good for us, for this study, okay? You always have to come up to, you have to develop a little bit of a series of rules of thumb, if that makes sense, and become a bit more selective. But it boils down to the questions that you're interested in, of course.
Right. Thank you.
Thank you.
So Rob, if you can, now I'm going to try to stop the YouTube transmission. I'm not sure if I can.